Jesse Poland is a research geneticist with the USDA ARS and holds adjunct appointment in the Department of Agronomy at Kansas State University. Dr. Poland's research group is located on the KSU campus with field research across the state. He currently supervises 3 graduate students and a postdoc scholar, and also sits on graduate student committees of 5 other students at both Kansas State and Colorado State University. Research in Dr. Poland's group is focused on wheat genetics and germplasm improvement. They're currently developing new marker technologies for use in breeding, diversity studies, and association genetics. In collaboration with public breeding programs, Dr. Poland is exploring the use of genomic selection methods in wheat breeding. In the area of germplasm development, Dr. Poland's group is focused on developing new breeding lines with resistance to the major pests of wheat, including stem rust, stripe rust, leaf rust, and Hessian fly. Dr. Poland's lab is developing high-throughput phenotyping approaches for field-based evaluation of breeding lines, with the primary focus being genetic characterization of heat and drought tolerance and development of improved germplasm. And today we're going to be hearing...
Thank you. And thank you for the nice invite to give the seminar today. And so you can hear me okay right now? And so I'll just start in with this and talk about some of the things we're doing for high throughput phenotyping, and then how this relates to plant breeding and genetic studies. And so really the goal when we think about biology and then also plant breeding is to really connect a genotype, which is a heritable, transmittable characteristic, and connect that to the phenotype, which is really what we're ultimately interested in these plants. And so really the way we do that, though, is we observe a phenotype and then try and correlate that back to a known genotype. And then from there, what's actually happening is the genotype is some DNA sequences that are transcribed and translated into proteins, and then these proteins work in various networks to actually produce that phenotype. And so really what we're doing is creating a more or less sophisticated model to basically connect that phenotype, or the other way around, connect the genotype then to predict predict ahead of time what the phenotypes will look like. And so just some example then, if we think about actually phenotypes predicting other phenotypes, or basically correlated traits, here's an example of a physiological yield prediction where canopy temperature is actually a very good indicator of overall yield. And so what this says is that for like correlated selection or traits that are easier to measure, those can be used for more difficult traits. And so now what we think about in this paradigm is we have phenotypes and trying to connect those to genotypes. But then we also have a range of correlated phenotypes and proximal sensing phenotypes, and we try to determine how those can then relate back and be used to model and predict more complicated or difficult to measure phenotypes. Also, when we think about genomic selection and training populations, here we're basically building a model using the genotype information on known phenotypes, and then using that trained model to do prediction. And so we do need inexpensive high-density markers, and these have been readily developed, but we also need accurate phenotypes to train that model. And so, so this is what we need to think about moving forward. Sorry, my slides are loading here slowly. So to think about what high throughput means, I think everyone is probably familiar with high throughput genotyping, and there's various levels of high throughputness. And but the real key with genotyping is that all of this wet lab work has been readily automated and becomes easily automated, and then becomes very high throughput, hands off. And so, and this translates when we think about the just the trend in genotyping versus phenotyping. So, for example, this yellowish line here is sort of a measure, and as things move towards sequence-based genotyping, the cost for sequencing is drastically dropping, and you can see this is actually on a log scale on the right. Whereas if you use some proxy for phenotyping, say minimum wage, if you have to get a bunch of undergrads out there to do hand measurements, or if you are looking at input costs to run field nurseries, those costs are continually increasing. And so if we're going to define then high throughput phenotyping, I think there's a couple things to really think about. First one here would be that it's fully or mostly automated data collection. And so what I mean here is that this can be then scaled up and automated, and so it requires less and less time as you go to bigger and bigger experiments. The other one is we need to really kind of define what amount of time to spend per entry. And so something like 1 second becomes a nice target where you can do thousands and thousands of plots over a short period during the day. Also, the big thing to think about too is that data analysis must be pipelined, in the sense of if we spend hardly any time collecting the data, but then the analysis takes lots and lots of times, it doesn't really become a high throughput system anymore. And then also we have to kind of think about the difference between high resolution and high throughput. And so a lot of phenotyping systems are really being built around the concept of high resolution, but on a per entry or basis, they're actually not very high throughput. You can only handle maybe in tens or hundreds of entries in that system. So to think now about like phenomics and just the power to translate observed phenotypes into understanding genetic effects, this is just a figure from the paper talking about the statistical power of the nested association mapping population. And just the take-home message is that the power to understand these traits comes from using really large population sizes. And so you can see that basically these red lines and trends here is that as you increase the population size to thousands, you really never see a decrease in the power to detect small QTLs. And so with that, we really need these developments in high throughput phenotyping to translate all of this genetic information, this genomic knowledge that's readily available. And so this is how we're thinking about high throughput phenotyping. It's really becomes a multidisciplinary approach. And so we sort of have this traditional plant breeding and genetics focus, where we're looking at a lot of entries in these design field experiments or design genetic experiments. But now we overlap with physiology in the sense of using different proximal measurements to define physiological parameters. And then to really truly put that into a high throughput system, there's an engineering component where these have to be integrated into high throughput systems. And then finally, this bioinformatics piece. This is really the processing of these huge amounts of data that can now be collected on these high throughput systems. And so here's just, I want to talk about some different platforms, but this is from the Australian Phenomics group, and just highlighting some of the different ways to think about um, analyzing and imaging different levels of field plots, and from the growth chamber and greenhouses and out through the field. And so with that, there's been a lot of effort and investment on these phenotyping chambers. And so this is some stuff from Limnatech. And these are really like fully automated greenhouse type chambers where these plants move around on these moving pots, and they can be fully imaged, very controlled, precise conditions. And so the good thing on this is they get really excellent detail in their controlled conditions. And it is fully automated, so you can do these large experiments with very little hands-on time. The downside is that these are limited space, so once you've sort of built one of these systems, you're really confined to the amount of space that was set up. And there's always the question of how these translate to field conditions. And also, they are quite expensive systems. So another one that we've looked at is this helium balloon. And so these are sort of like weather type balloons, and you can mount different cameras on those. And so some advantages of this platform is that it's pretty inexpensive, it's low tech, so you can deploy it pretty easily in the field, and you can start to image whole fields at one time. The downside is it's helium, and actually turns out that you might have heard of global helium shortages. It turns out it's kind of hard to get helium. And also it turns out it's not very good in wind, which is a problem here in Kansas. And there is a limited payload. And so you can only put so much sensors and cameras on this type of platform. So some other stuff that we've been working with is this remote control helicopters, or these planes. And so here's just some pictures of that guy. And I'm not going to talk about this much today, but you can see some images here on the right using false color images and just regular um, uh, red-green images here. And so with these, you can image entire fields, and they're also not too expensive, and they're pretty flexible on the deployment. The limitations on those is you do need somebody who really knows how to fly the thing. And then also they have a pretty limited payload too. So you're sort of limited to one, maybe two cameras that you can take up in this type of system. And then, so you can really develop some pretty nice detailed imagery from that. And this is actually a 3D mosaic of multiple images that were stitched together from that little helicopter. And so you can see you can really start to define the plot pretty well. And so this is a good system, and it's under development. So what I mostly talk about today is our work on these phenotyping vehicles. And so some advantages on those is you can carry lots of equipment. So we sort of have a really large payload. So basically any type of sensors that you want, you can put on these. They're pretty flexible deployment in the sense that we can get out into the field about any time, and they're easy to operate for guys who are used to doing the field planting and tractor driving and things like this. And so on the downside is you can't assay the whole field simultaneously, and then they are not completely automated yet. So with this, we're using this idea of geo-referenced proximal sensing. And so I'll talk about this some more later. And we have a number of different sensors: green seeker, infrared thermometers, sonar sensors. And so I'll talk about these more in detail. But I think the real key is to note here that we want to physiologically define the proximal measurements. And so this is where the physiology piece comes in, is if you can look at these studies and say, oh, canopy temperature has a large impact on yield or plant health status, we can then say, okay, well, how can we measure this on this high throughput platform? And so with the geo-reference, this is really where precision ag meets plant breeding. And so really what we were doing here at this point is taking a lot of these precision ag tools and then translating them back into small plot research. And so, for example, all of our plot planting is done using a GPS, which trips the plot planter. And so you can see in this picture here how it's very easy to put in really precision planted nurseries. And so with this idea of the geo-referenced proximal sensing, here's a little cartoon mock-up of the phenotyper. And basically you have this GPS unit, and then you have sensors. And based on this GPS position, you know the position and the points where you're collecting all of these different measurements. And then this just goes into a data logger, or now we're actually using a PC for data logging. And so the platform we're working on most recently has this configuration here where we actually have 2 GPS units, one on each end of a front-mounted boom. And so I'll show in the next slide how this is used to take the orientation of all the sensors. And then we have a number of sensors mounted in multiple row configurations here in the front, and then a computer up here with the GPS units that's logging all of the data. And so on each one of these sets of sensors, the picture here on the right shows that the multiple sets of sensors that we can run on this thing currently. We have a green seeker, this crop circle, a sonar, and an infrared thermometer. So the IRT can measure like canopy temperature. The sonar can measure plant height. And then the crop circle and the green seeker are active sensors that can measure things like normalized difference vegetation index, or NDVI. And so with these 2 GPS, we can keep track of the orientation of that front toolbar and then all of the sensors because we know their positions relative to the 2 GPS. And so what I'm showing you in this little graphic here is the GPS coordinates of each of the sensors. I'm sorry, of the 2 GPS, and then the 3 sets of sensors between those 2 GPS. And this is just a small subsection of a pass across the field. And so what you can see is sort of this would have been driving from the left to the right and then starting to turn around and go back for the next pass. And so you can see that the NDVI values are colored based on each of the sensor readings. And so here you can see basically the different plots, and then you can see the alleyways here. And so with these two GPS and then the multiple sensors in the middle, we can always keep track of the orientation of the platform and then precisely place each of the measurements between those 2 GPS. And so this system is really a turn it on and go type of platform. And so this is just an aerial satellite image of the agronomy farm where we do a lot of this work. And so here's one of our field nurseries up here at the top. And so what I'm showing here on all these red points is basically a 1 day of data collection, where they pulled we pulled the phenotyper out of the shed here at the bottom, turned on the sensors, and then drove down the road over here over to the field, and then drove back and forth up and down the passes on that field collecting all the data. And so with that, if you zoom in on that field, you can see then with each of these positions, there's some 10,000 or more measurements here, so you can't really see the individual points. But moving along the path here, you can see the path that the phenotyper took and then referenced on the coordinates of each of those points. And so if we take that same field and then we color each of those points based on the NDVI value, you can start to see differences across the field. And so you can see kind of field level variation where this top left corner has lower NDVI values. That was sort of a poorer soil that was drought stressed there. And then you can also start to see pretty big differences between plots. And so if you look here on the left, each one of these kind of red lines going down are the alleyways between plots. And so, and then each one of these small sections would be an individual plot. And so now what we can do based on this and the outline of our field, we can assign those data entry to those raw data to field entries based on the coordinates. And so showing here on this slide is if we zoom in on a small section of the field and we look at the raw data, you can see the continual sensor measurements that are coming in as we take each pass up and down the field. Based on the plot coordinates, we can define the 4 coordinate boundaries for each one of the field plots. And then based on those coordinate boundaries, we can trim off the data that is basically in the alleyways. And then based on our row column design of the field plot, we can assign the data for each one of those plots to an experimental entry. And so you can actually see on this bottom, on the bottom right here, you can actually see the pretty measurable, sizable differences in the NDVI value between these different plots. So, and here's just another example of plant height measurement. And so what I'm showing here is just the data from a single pass down one column. And so what you see is the sonar is basically measuring the distance from the toolbar sensor to the top of the canopy or down to the ground, whatever it will hit first. And so you can see each one of these measurements then as the vehicle travels along. You can see here measuring the ground on these lowest values and then measuring the crop canopy on each one of these different colored lines right here. The other thing with this GPS is that with the RTK correction, it's actually accurate enough to go back and assay the same plots over and over. And so what I'm showing here in these multiple different colors is actually data that was taken at 4 different time points. And you can see that we're actually able to overlay each of these measurements directly on top of each other. So this actually enables then multi-temporal measurements. So we're able to go back in this high throughput manner and start to look at the same fields over and over and look at dynamic changes throughout the year. When you look at these differences then on a genotype or an entry basis, you can start to see these dynamic differences throughout the growing season. And so here, for example, at this first, here, this is just the NDVI curve for 30 different entries from the advanced trial. And you can see that most of them start pretty close to 0.6, and then they pretty all uniformly end up at around 0.1. But between those two time points, there's actually a very dynamic difference in the rate of senescence on these different genotypes. And so we can now start to look at those differences. And so I've been mostly talking about this phenotyping mobile unit, or the PheMU, as my guys affectionately like to call it. And so one thing we've envisioned is if you take this PheMU and then you combine that with a unicorn, then we come up with this phenocorn. And so this is basically a handheld version of this mobile platform. And so just to briefly talk about what this mythical phenocorn creature looks like, is basically the same setup in a handheld device. And so it has this a GPS unit here for referencing all of the data. It's fitted with an IRT and a green seeker. And so this is Jared Crane, one of my graduate students, and down at CIMMYT, Mexico, taking measurements on some of his experiments there. And so you can see here on the right the data coming off of this platform. It looks very similar to the data that we collect on the mobile platform. And so we can basically then start to apply the same algorithms and tools for translating that raw data into observed phenotypes. Also, just to point out a few other configurations that are being developed out here. This on the top two, these are photos from the group in Maricopa, Arizona, who we've been learning a lot and collaborating closely with. And so sort of a push cart type here with the GPS here again, and then cameras and sensors in the front. And then a similar setup for cotton with the mobile phenotyping platform here. And then here at the bottom is a system that's being developed at Erie for rice. And so you can see this really long side boom, and where you can actually drive this tractor between the rice paddies and then hang this boom over into the flooded paddies and take the same type of measurements that way. So really, just to briefly touch at this point, just thinking about then taking these multiple layers of data and measurements, and then how do we really move this into physiological understanding of the phenotype. And so this is really where you get into multiple different layers of plant modeling and converting these basically proximal measurements into plant characteristics of interest. So something like taking NDVI and then from regression, getting leaf area index out of that. And so also with that, then you can start to envision more complicated but holistic models, taking into account all the layers of data, but then also soil and management and weather type parameters to basically model crop growth throughout the season. So now just to come back and think about connecting that genotype to phenotype, and we talked about this earlier with observing the phenotype, and now we've really started to develop a high throughput system to really observe these phenotypes, and then how do we take that further and translate that into a better understanding of plant growth and development on this temporal scale throughout the season. And so now I'm going to shift gears a little bit and talk about a kind of a different thought on high throughput phenotyping, and this is what we can call massively parallel. And so if you think about a HiSeq 2000, this is one of these next generation sequencing machines. It's not that impressive if you think about it, that it takes 5 days on this machine to sequence 100 base pairs. So if you have a 100 base pair fragment, it takes 5 days to actually determine that sequence. However, it does 1.5 billion of those sequences in parallel. And so this is how these sequences are such high throughput, is not because they're actually working that fast, but they're actually doing so much in parallel. And so if we think about this in phenotyping world, we can maybe start to think about some massively parallel phenotyping. And so this is just a little side project that we've been working on and thinking about some on-farm testing. And this is again employing those precision farming tools, precision ag tools that are really becoming ubiquitous across production agriculture. And so this is actually a little project. I talked to this is my father on the right and my uncle on the left. And so I talked them into this little project last year, and we're repeating it this year with another farmer. And so this experiment was 2 replications of 7 varieties, and then a commercial blend that was in the rest of the field. And that blend was these 3 varieties, Everest, Art, Armor. And so these are actually planted in strips, and they're 35 feet wide by 1,200 feet long. So that comes out to almost 1 acre. And we could randomize these. And the way this was done was to put 1 variety at a time. And then because these are planted using an auto steer, auto guided tractor, you can actually plant 1 strip down the field and then skip over a number of strips and plant another one. The blend was planned as a check, and then we can harvest and record these on plot weight. And so the really interesting thing that starts to make this amendable is that these onboard yield monitors have become very accurate. And then to check this, we tested the onboard monitor against a seed tender, a cart weight. And so what this looks like planting these strips, this is some ones we put in just this last fall. And so you can see that these huge air seeders, and these are planted in big strips down the field. And then you can actually skip over a number of strips and make the return pass, and then fill in between those using the auto steer. And so then when these are harvested, you can just cut a strip right out of the middle and basically measure the complete weight, as well as taking the yield mapping, the yield across that field plot. And so these onboard yield monitor measurements were then checked against a weigh cart, and so that's what we're doing here. And then this gave sort of a concrete measurement of, or basically a check against basically the real-time onboard yield monitors. And so what we found on this was that the yield monitors were actually extremely accurate. And so you can see here the correlation between the cart weight on the bottom axis and the combine weight on the vertical axis. And so this correlation is like 0.99. And so to note that this combine weight is basically taken in real time, and so there's no measurement. This would be at the speed of irregularly harvesting the field. And so we had this in 2 replications, and so there's actually a really good correlation between the yield for the 2 different reps. And so shown here on the bottom is rep 1 and then rep 2 here on the vertical axis. And so you can see the correlation between the 2 yields observed across those is really good. And then based on that high good heritability and correlation, you can actually see that we're able to tell pretty pretty accurate differences between the performances of different varieties. And so that was good preliminary testing to see there. And so what does this look like then if you envision about scaling this up? And then it really becomes like a crowdsourcing and truly massively parallel endeavor, is that if you start to envision a network where hundreds of different farmers are actually growing a few entries each, then across this network, you can start to evaluate tens and hundreds of different entries at a time. And so, and then the cool thing is these plots are so big that you can actually image and look at this remote sensing data from the satellites that are routinely flying overhead. So I just have 2 acknowledgement slides here. Here's some of the key people for all of this work. Up on the right, up on the top left here is Jared Crane, and then here on the phenotyper is Josh Sharon, and driving the phenotyper in the middle is Ryan Steves. These are 2 of my technicians here. On the top right is Jed Barker. He's an engineering student who's really been instrumental in developing our current platform. And then my father and my uncle there on the bottom, George and John Poland. They were good participants in the massively parallel phenotyping study. And so here's the full group of acknowledgements, and really want to point out the group in Arizona. Pedro, Mike Gore, Jeff White, Kelly, and Andy, really pioneers, I think, in this this phenotyping work. And so we've enjoyed collaborating with them and have really learned a lot. And so the support for this work comes from USDA. We have a new NSF project to develop the phenotyping platform and analysis tools, and then also from the Kansas Wheat Alliance. And so this picture here is just taken actually from the remote from the helicopter, and this is the aerial shot of our phenotyper. So I think that's all I have for now, and I'll take any questions. So Jamie is asking, how have we automated analysis? And we're getting there. So really with, and that's a lot of what our NSF project is going to focus on, is actually taking this raw data and then developing pipelines to turn that into data that's basically ready for QTL mapping or for any type of normal analysis. And but really, once we define, so for example, on the ground-based platform, once we have the plot coordinates, it really becomes a pretty simple query to basically join whatever data comes from within those plots to that experimental entry. And so, okay, so Dave Matthews asks that he says he's seen some satellite images with resolution near plot level, and what are the possibilities with that? So I think that isn't actually a good possibility. The trouble with those, so with satellite images, there's two things. So the satellite, you can envision the satellite is either taking a picture of a really small area on the Earth's surface, or it is taking a much larger area, a much larger picture. And so, and then there's a return time on how long it takes that satellite to get back to that same spot. And so obviously if it's taking a much larger picture size, then it returns to the same area more frequently. And so a lot of those really high resolution photos that we get from like Google Earth are actually, they only return to that same spot maybe every few months. And so it really becomes then whether you're lucky enough to get those images at the right time in the growing season. And so, but I think definitely, I mean, the satellites are only going to get better and better, and so I think this is the way it will move in the future. Any further questions? How much does a phenocorn cost, and will you share the design? That's a question from Jamie. Sure, we'll share the design. I think the phenotyper, it depends on what type of GPS, and so the different components probably come out to about 10,000 total. And so, but we need to do some more experimenting with the GPS is actually the most expensive component, and the reason being is that we need a GPS unit that has a differential correction, like RTK or OmniStar, so that you can get down to a GPS with plot level resolution. So something like the GPS in your phone is only accurate to say like a few meters, and the GPS that we are using is accurate to maybe a few centimeters, 1 or 2 centimeters. And so with that though, we've basically developed this platform and the associated software that drives and records all the data. And so as part of our project, we're going to make that all available. So stay tuned, and it should be available pretty shortly. And so, yeah, Mary asks about the details on the GPS to get this level of precision. And so we do have to use some differential correction. And so this is like OmniStar or RTK, which is real-time kinematic correction. And so these will give you, and these are the expensive GPS units, but they'll give you resolution to like 1 or 2 centimeters. Oh, and Sarah asked if the phenocorn is really heavy with so many instruments on it. And I think it's fairly heavy. I haven't got the opportunity to carry it around yet. But, and so we've got a little design work to do to make it lighter. But also a lot of these groups have been carrying around a similar instrument, just without the GPS and the PC for data recording. Araby asks, at what point in the breeding process is this type of phenotyping meant to reduce costs or time? And so, yeah, so this is sort of an open-ended question, I think. Where I would really envision it in the breeding process would be at the first year unreplicated trials. And so, particularly for wheat, a lot of programs work this way. And so this would be the place where you might be able to take a nursery
Thanks for
from
your
maybe
presentation today.
1 or
It
2,000
was really
entries to
interesting
10,000
and
entries.
had
And
quite the
so,
lively discussion here.
recording something like heading date on that many entries becomes a real
this is the last
real limitation.
TCAP
Basically
webinar
taking any
of the
measurements
spring
on
semester, so watch for
also, you can
announcements
start to
in the
envision
fall
this too
about
as taking sort of
the
multi-temporal
fall
measurements
series.
that would not really be feasible at all with if you had to take the measurements by hand.
Pointed out we need
Another big
volunteers
application you can think
to
about in breeding
help
is for non-destructive
organize
biomass
the fall series of
sampling
webinars.
estimations.
So if anyone would like to volunteer or volunteer somebody to speak,
and so with that,
I'm a good
if you
contact
have these
person
different parameters, you can actually estimate
for
biomass, and then
letting
you don't
know. You
have
can
to
shoot
take
me
a
an email at
destructive
that address
measurement
there.
of the plots. And so for forage breeding programs, that would be a pretty big benefit. Also, you can take this type of measurements a lot faster and high throughput relative to actually doing a full plot harvest and a biomass weighing. So Mary asked about the
well, thanks,
NDVI
everybody.
it saturates. We don't really